Today we are discussing about the learning and memory, the physiological perspective. Now, I have a figure here on the right hand side showing the brain and the medulla and the spinal cord. So you have the four brain areas: the frontal area, the parietal area, this is the central sulcus, and then this is the occipital area, and this is temporal area. Now, you know that this frontal area is to remember and pick up. These are trying to know about the facts, dates, etc. Then this is about what you see around, and this is about what you know about your phone numbers or phone call. Somewhere here you have the auditory system. This is temporal area and the today's lecture I have planned in this way. One, I will define the learning and memory. Number two, I will tell you about the types of memory briefly, and I will tell you about the brain areas and or centers which record memory. And then fourth, I will tell about how the memory is formed. Fifth, I will talk about the neuronal and molecular basis of memory. And sixth, I will talk about the disorders of memory. And seventh, I will mention about the memory enhancers or memory increasing points, especially the physiological basis of memory enhancers. Okay, so now, having seen the plan of the lecture, so start with the definition. Learning is a process by which we acquire knowledge around the world or about the world. That is learning. The memory is the process by which the knowledge is encoded, stored, and retrieved as required. That means one is acquiring, another is putting it in a coded form and storing and then retrieving it as and when required. And both are interrelated. They cannot be separated. We have two forms of memory, the explicit memory and implicit memory. Explicit memory is also known as a declarative memory, wherein you express. It is also known as autobiographical memory. Okay, so this explicit memory has two components. One, which is associated with the facts, description, concepts, and grammar. That is a semantic. That's concerned with this. That is a semantic memory. Then we have a episodic memory. This episodic memory is about events, places, the associations, whatever linked with you or your surroundings. Then I think that is very clear: declarative memory, semantic, and episodic. Semantic is facts and the episodic is events. Implicit memory is a non-declarative memory. There are four components in the implicit memory. One, procedural. Implicit memory, which is procedural. This is the motor skills we develop during our life. Just whatever, cycling or motorbiking, racing or swimming, or whatever motor skills we develop. Maybe tomorrow you would become a great surgeon, and that is the motor skill you develop and how to perform a particular operation, something like that, particular surgery. So then that is about the skills you develop. The skills may be developed by you don't require the explicit memory for developing a skill. You just know the carpenters, the masons who build these structures. They may not have that particular explicit memory, but they are procedural memory is very much powerful. Then the habits. In the habits I mentioned about the tapping a table while listening to a music. because we are automatically associated with that. Or what happens when you start listening to the music or particular event, you start tapping your legs. So on, we have certain habits developed along with each of them. So that is the habits. So I have just given two examples. Then priming is a event in which we learn or we get the information by certain important clues. So these clues are just I have mentioned here ILU. Other day I was talking about when this ILU is there, you just read it as I love you. That is, or icons. Nowadays, so many icons are used in our communication, especially the Facebook or the WhatsApp communications. We use those icons. The symbols we use. Here, I have just given you uh, what you read here. You read ambulance, though it is meant for a mirror image, but if I see these words, those I have reversed the entire word, I read it as ambulance only, because I will not see EC and whatever, something like that. It is an ambulance. So that means this is what the priming is. So already we are primed about ambulance and how the ambulance is spelled, and we try to reverse, our brain try to reverse it and try to give you that. That's priming. So now for you, I don't read for you. So this is what the new culture has developed with the the mobile messaging system. Then we have associative learning. The associative learning we talked about are it is associated with the conditioning reflexes, especially the conditioning components. One, there is a classical conditioning of Pavlov, and then the operant conditioning by Skinner. So that means we have associate learnings associated with Pavlov's classical conditioning components, or the operant conditioning operated for reward and punishment. So that is a Skinner's experiments. Then we talked about the sensitization. Sensitization is a term used in the implicit memory that the particular person is trying to be sensitized for this thing. Suppose I was sleeping in a strange place. There is some time clock which is producing the tick tock sound, and that tick tock sound becomes loud and louder as I try to speak further. Sometimes it may happen to the people. So that is sensitization. The more you think about it, and more it louder you hear. Something like that. But another important example, the one example I gave now, TikTok sound when you are sleeping in a strange place and you are trying to get that. Or a newborn, classical example is a newborn baby's cry. This is a very friable, a very low, but the mother will get up and respond to the newborn baby. So that is desensitization. She may not get up with other sounds produced. Then habituation. I was talking about habituation. That means habituation. What happens whenever we are taking the class? During the class, you just doze. You will be in the entirely different world. Maybe with a NREM or REM sleep. So like that, dozing. That is the sort of a habituation. That means the habituation here we means desensitization. Desensitization. So that means the dozing during the class, that is a monotonous work, or sleeping in a noisy place where there are noisy places. I mentioned about a train journey where a train will produce a enormous amount of noise, and then still we are able to sleep. So these are the types of memory. Try to remember these things. I briefed about the types of memory. Then we go further. What are the brain areas or centers for memory? Now let us consider what are the areas for explicit memory. Naturally, for the explicit memory, hippocampus, here is the hippocampus there, this is the hippocampus, and dentate gyrus there. This is hippocampus. Then amygdala, this is amygdala. Hippocampus is the main this thing. Amygdala is associated with the emotions, emotional components. Then we have a thalamus here. This is basal ganglia. Somewhere here we have a thalamic component. So then hypothalamus, that is also required because a mammillary body is required for memory formation. Then we have a basal ganglia, that's striatum. So these are the things required for explicit memory. For the implicit memory, so of course here I have not included, maybe I will include prefrontal cortex also, prefrontal cortex also for explicit memory. And I also include the cingulate gyrus, somewhere here. This is the cingulate gyrus, this green area, prefrontal cortex. So all these things are necessary for explicit memory. Then implicit memory, the implicit memory is about the skills and the procedural components, or developing those associated learning. That means classical conditioning, all those things. You require a basal ganglia, the cerebellum, the motor area, especially the sensory motor area. You require these three components. That means you require a basal ganglia, you require cerebellum, and you require the entire sensory motor area. Especially the sensory motor area is here. That is the motor cortex. This is the primary motor area here. But the motor for corticospinal tracts originate from the M1 motor area, then premotor area, and some part of the somatosensory area, S1. So that is why I'm written sensory motor area or excitomotor area. That is all are involved for the implicit memory. So then for working memory, we require prefrontal cortex and basal ganglia. Not only because it's primarily prefrontal cortex, the working memory, it is only very short time, less than a second. You forget about it, and it is just storing and then forgetting, because we don't have a space in the brain to retain all the information, because you are every now and then you get a number of information, you have to filter them and you have to discard certain information. So this is working memory, so that is only for a short period of time, so that is processed in the prefrontal cortex. These are the areas of the brain. So I mentioned what are the primary: hippocampus, amygdala, the prefrontal cortex, the number one, hippocampus, amygdala, prefrontal cortex, and all the sensory areas. All the sensory areas. I will come back to that. So then we have a thalamus and hypothalamus. They are also involved in the memory. Implicit memory is primarily motor, the basal ganglia, cerebellum, and sensory motor area. Working memory is a function of prefrontal cortex. Now we move on. So I have already mentioned about it. Is associated, hippocampus is associated with short-term memory. Not only short-term memory, it helps the encoding, consolidation, storage, and retrieval of the memory. The parahippocampal portions, medial temporal cortical areas, they have the circuits, so they will connect to the entire neocortical structures. That means the parietal, occipital, temporal for auditory and olfactory areas. So to process these information, all these sensory cues, so they are also involved. So that means along with the hippocampus is attached with the sensory association areas, and these sensory association areas are linked with the hippocampus, and they will try to encode, consolidate, and store and retrieve the information. Now, amygdala is closely associated with the hippocampus, and this amygdala is emotional center. So that means it is in all the information regarding emotions are stored here in the amygdala. So that means you try to remember a snake. So there is an emotion associated with the snake. That means a fear or withdrawal. So that type of emotion. So that means the amygdala is activated during that time. Or anything, a sudden fire there. So that means amygdala is associated. So that means that type of all the emotions, the whatever the type of a positive emotions or negative emotions associated with a particular thing, they are stored here in the amygdala. Then medial temporal cortex in association with the hippocampus, they are processing the short-term memory. So the short-term memory is with all the sensory modalities. Prefrontal cortex, working memory, thalamus, hypothalamus, short-term memory and working memory, sensory association areas, they become a primary seat for storage of information for long. So that means hippocampus cannot store or stock all the information. So the hippocampus gives the responsibility to the associate particular association areas. That means if it is a somatosensory, so it will tell that, come on, you store your information. I will ask you whenever I want to know about a particular event. So that means the final storage of a particular sensation, either it is a cutaneous sensation or a visual sensation or olfactory sensation or auditory sensation, they are stored in the respective association areas. These are the brain areas of the memory. So now I just again just tell something about the hippocampus and the thalamus are involved in the memory. So these connections are very important. So why they are important? I mentioned about a Korsakoff's syndrome, Korsakoff's syndrome, in which the mammillothalamic tract is involved. So there is a loss or a demyelination of the mammillothalamic tract. So some people with alcoholism related brain damage develop impairment of recent memory, especially the recent memory, and the memory loss correlates well with the presence of pathologic lesions in the mammillary bodies and the mammillary body to thalamus, anterior thalamus, the mammillothalamic tract. So they will connect the anterior thalamus, and there are lesions there, and that would produce the disorder known as a Korsakoff syndrome. There is a loss of the recent memory. What's called an anterograde amnesia, or he cannot form the new memory there. So now this is because the fibers concerned with the memory project to the prefrontal cortex and there to the forebrain, basal forebrain. So now this is one of the important thing, especially from the nucleus basalis main heart. In the basal forebrain we have this the basal nucleus of main heart, and this is necessary for the processing of the short-term memory. So in case of Alzheimer's disease, there is a loss of neurons of the nucleus of main heart, or the circle of main heart. These cholinergic, because it has a cholinergic neurons, these cholinergic projections are lost, and once those cholinergic projections are lost, there is a loss of short-term memory, so that they are connected with the amygdala and the hippocampus. So I just give you a brief idea about how in Alzheimer's, I have given two diseases: the anterograde amnesia, which is happening with the Korsakoff syndrome in the lesions of the mammillothalamic tract, and in the lesions of the nucleus basalis of the main heart, or circle of main heart. So we have the loss of the short-term memory because this is not connected. The neocortex and the amygdala, hippocampus are not connected. There is a Alzheimer's disease. Then how the memory is formed. So there are three important things to look into, to consider into the memory. One, the memory, as you have seen, there is no single place for which the memory is stored. The memory is stored in multiple sites. Point number one. Now, the memory has multiple associations. So, for example, if something is there, say, for example, even I mentioned about a snake. Snake, there is a visual cue. That means you know that this is a snake. So then you will know that how it produces a sound. That is the auditory clue. Auditory clue. So that means the hissing sound of the snake, you imagine immediately. That means that attribute will come back whenever you recall the memory of a snake. So then you come back with the other sensory clues like that. If it bites, it produces pain. So that means yes, that is somatic, these things. Then fourth thing is you know that it is deadly. That is the emotional aspect, the affective component. So all these things, that is what I was trying to tell in one. They have each one of those memories have four attributes or five attributes together. They are processed. So that is how the brain stores multiple representation. If I give one, you will be able to connect to the particular. The his I say, you will bring the snake. Or if I write something like this, you will bring the snake. So, or if I tell somebody is dying because of the bite of this thing, you may think first the snake, something like that. So the memory has multiple representations. So there is no single place to store. It has a multiple components. Then, so the memory formation involves the following steps. One, the steps in the memory formation. The second, processing sequence. Now, steps in the memory formation, the steps are sensory memory, less than a second, short-term memory, minutes to hours, working memory, less than a minute, even less, and long-term memory remains there for hours and days and sometimes entire life. Maybe your association with me, you would remember me even uh, after maybe your whenever you meet, you have your own class people meet, and then you try to remember your teachers. So yeah, as we did in our golden jubilee celebration, we remembered our teachers, because that is the type of long-term memory that is remaining for long. So now this is four components here. So another four components here, how the memory is processed. It is processed in a sequence. The encoding, that is a codification, then storing, that is a consolidation, or that means before storing it has to be consolidated or it has to be processed. That processing is called as a consolidation. Then after the processing, it is stored into the neural elements, that is the storage. So then with these, then there is a fourth stage, because the memory once stored, if it is not retrieved, then it may be lost. There is a retrieval. The memory is retrieved as for our usage, as for our demand. So these are the four things. So that means all these four things, what I am trying to tell. So either the steps or the processes, they happen repeatedly to form a memory. So that means each are interdependent. The steps A and steps B are interdependent. They are necessary. Say for example, encoding, sensory memory, short-term memory that encodes. Then after that, these things will consolidate. So we'll go to this next stage. So now the steps of memory formation I just mentioned. The sensory memory, it is the sensory components coming from olfactory, optic, auditory, somatic, and other sensory modalities. All these things are uh, the memories. Suppose if I, yeah, if you happen to, if I am asking you what scene you have watched, you may just tell briefly, just I saw a hillock, or I saw a hill, something like that, or whatever you have surroundings. But that will not be stored forever. That is only for remains there for momentarily. Then short-term memory, so that is staying for minutes to hours. during which the processing in the hippocampus, because if this short-term memory, if you feel that it is relevant or it is helpful for your advancement of knowledge, so what happens? The hippocampus takes it on and takes it on further. If you feel that this is just okay, so as we watch TV, sometimes we watch TV, the news items, some of the news items we watch and we forget, we don't care, we are not interested. Some of the news items we try to remember, so like that. So the short-term memory is there for minutes, and then according to the relevance of the particular area, we just hippocampus decides whether we want to take it or you don't want to take it. So then it will go for the changes in the synaptic transmission, so that I will come back. So that means there are long-term changes in the brain areas that would see that that particular information you are interested, they will be stored. That is a short-term memory. So now from this short-term memory, it enters into the long-term memory. Now there is another memory, what we call it as a working memory. Working memory is again a short-term memory. It's another form of short-term memory, and that is trying to remember. So say for example, I mentioned that I have gone out and trying to have some information about a place. They will say that this market is here, this market is here, and I am trying to locate that, how much is the distance of that, how much is the distance of that, and that is a very simple thing we try to remember and then forget. So this is a working memory to perform particular action. So that is a prefrontal area. So that is a this is exactly planning of the particular or execute particular work to be executed. So that is a prefrontal dominance or prefrontal cortex is involved. Long-term memory, which stores the information, and this stores the information permanently. In the beginning, it begins with the hippocampus. Later on, hippocampus says that it will give it to the respective sensory areas and to retain. And now the hippocampus tries to retrieve it from the old memories and trying to bring back, so that this will remain for long. Now, started the environmental, this is our sensory inputs. These sensory inputs forms the sensory memory, and this sensory memory with the attention, that would become a shorter memory. So now this shorter memory, so that means it will be thinking about with I mentioned whether with the hippocampus, already it is hippocampus is registering the shorter memory and thinks that whether it is worth retaining or not retaining. If it is not retaining, it is forgotten, it is rejected. If it is worth retaining, it will try to be rehearsing. So that means in the mind, this starts rehearsing. So now this, after elaborate, after several rehearsals, it will enter into a long-term memory. It is stored in the hippocampus there. So now the hippocampus try to retrieve it, and these cycles go on. So that means the first cycle is here, rehearsal cycle in the shorter memory itself. So that means you remember when you started learning the tables, how many times you repeated, how many times your parents or a mother asked you to repeat those tables, which we really the Indian parents try to enforce upon. So that means you repeat, finally one day you will become proficient in telling. That is a rehearsal. So once you make the rehearsal, when you become confident, then it is going into the hippocampus and stored. So that means the next day you start beginning. So this whatever stored information is bringing, and you make some mistakes with that again table and then corrected. So that means this whole sort of a cycle continues, and the memory retention or memory storage is taking place. So that means it starts with the sensory component, and then short-term memory, then it thinks about the importance of that, whether to accept or not to accept. If it is not accepted, it's rejected. So now this goes on, this cycle goes on with the long-term memory and the hippocampus. And if it is successful, suppose if it is relevant, you will try to retain it, and again there are some cycles of rehearsals, and this will end up in making the permanent memory. So that means I'm not talking about rotting or memorization. The memorization is an extremely difficult thing. I am talking about learning here. So that means we learned certain things not by memorization. I try to remember things in relevance to the number of sensory domains. So now once it is stored, this information is retrieved. So that means from here, this is retrieved, and then it has been given these things. These are the steps of memory formation in terms of the steps. Now go with the next component. So I mentioned there is a stages or a processing sequence. How the processing sequence of the memory formation. If you are talking about, this is how the stages are happening. So that means from the sensory memory to the long-term memory, sensory memory, short-term memory, working memory, and long-term memory, how they are processed. Now in this thing, what are the various sequence or stages? In this, we have four components: encoding, consolidation, storage, retrieval. The encoding, that means now I start. The start is here. Start with the sensory information. The sensory information today here is the lecture class on the memory. I gave a lot of information about the memory and the definition of the memory and all those things. This is the sensory auditory information along with the visual inputs. You have those class slides. Now this will be registered. So that means they will be registered in your brain. So you hear, you see, that means the auditory, visual, along with that association of your teacher. There are three components putting together. So that means they will try to encode. That means they will try to or code these things in this thing. That is the first step in the formation of the memory. So then once it is encoded, so that means that is processed to be stored. That is processed to be stored. So that means it is something like a vegetables. You want to make the curry. So that means you take the vegetables and you cure them. The encoding is a curing of that, and after that you consolidate them, you cut them and put them in the whatever cooking vessel, and then that will consolidate, and finally you will have the cook. It is a cooked material. It is stored there, and you bring it back and you eat it. So that is how the entire the memory is talking about. If I were to think about the memory as a curry we prepare, or a vegetable preparation we make, or any menu we try to make. So now encoding, then consolidation. Consolidation is a process by which the memory which is encoded, which is processed, and this processed information now is ready to be stored in the appropriate places. I have already mentioned what are those appropriate places. But the hippocampus and amygdala, they play an important role. Then after that, so once it is stored, there is a retrieval. Retrieval is that means it goes into a storehouse, and from the storehouse it is retrieval, that is taken, revived, or it is brought back to the surface. So that means it comes back with the consolidation stage or encoding stage. Now what happens here, I have just mentioned. For encoding, what do you require? You require attention. You require attention. So say you have a sensory information here. These sensory information are coming. So that means the sensory information is the whatever auditory lecture I am giving. Then the visual impact of these things. Then third thing is my own, you see myself there on the other screen. So they put them together, that would register. That is, if you are attentive, so then that encoding is this thing. Then motivation. Motivation is another important thing because you are now highly motivated because you have an examination coming up and you want to pass your examination. That is the most the strongest motivation. So you have a motivation, so then this encoding becomes easier because you have a positive approach for learning these particular whatever the topic or whatever the theme. So now the motivation. Then after motivation, rehearsing. Because if you hear my lecture and you just go home, go, and then after some time you don't remember anything, I swear. So that means you have to go and read and rehearse. So whatever the PowerPoint presentations I give you back, you just try to rehearse them the way I have told and try to understand them, because finally it is your brain has to process, not my brain. So if your brain has to process and store, you have to rehearse it. So that means rehearse. Rehearsing, again, I tell you that there are two components. One, rehearsing by reading. The reading has two components. One component, if you are reading silently, that sometimes you are lost because your mind is very fluid and it is wandering all the places. So now, if you are rehearsing loudly, there are two components. One, you say it loudly, the vocal cords are activated more, one point. The second point, you have a, what is called the sound produced that echoes back into yourself. So that means rehearsing, if you want to do, you do it loud so that it will be encoded by two times, one thinking and one auditory inputs. So when you are doing this thing, attention, motivation, and rehearsing, you need to continuously you start doing it. The whole thing becomes monotonous. It does not allow. So spacing or timing or intervals, you keep intervals so that encoding becomes perfect. Perfect. So that means you have a attention, you have a motivation, you have a rehearsal, that means a repeating. So then you give some space. So that means you stop for a while, you think it otherwise, then come back to it, then it becomes very well consolidated. So then we have a fifth component, cues. Try to associate a particular learning with some examples or with some mnemonics, with some other aspect, that is a cues. When you take that, so your learning becomes, or encoding and consolidation becomes much easier. So these are the three or four components. So then you also associate emotions associated with the particular sensory information. If it is emotionally involved, if amygdala is involved, so then you remember that particular learning forever, because whenever that type of emotion comes, it is easy to these things. These two components, especially the cues and emotions, they are very much essential when you want to retrieve them. When you want to retrieve them, so the way you have used the cues, so they will help you to get that particular type of information stored elsewhere easily, because now that is bringing from the stock or the memory roles. So otherwise the brain has a limited space and there are a lot of memories associated. So that means cues is a string. It gives us, it acts as a string to pull it. So that is one thing. Similarly, the emotion. So that means these two are helpful when you are trying to store certain information in the memory. Now, coming back to the consolidation. Consolidation, I have already mentioned the spacing. That is, you cannot consolidate at one go. On one day, you sit and try to learn about a particular thing. Not possible. You have to do it on repeated occasions, maybe 2 or 3 sittings or 2 or 4 sittings, depending upon our own mental ability. Some of them remember fast, some of them slower, but both are equally good in storing the information. So that means you need to store, you need to have a space or a time in between. Then again, on that day, maybe tomorrow again, if you rehearse, it becomes consolidated very strongly. So that means the consolidation becomes stable. So then that means some of the things exercise. There are two exercise. One, the exercise in terms of the mental exercise. First, I talk about mental exercise. You take the particular learning, and this learning you try to exercise or trying to bring the examples and try to retrieve that information. So that is the mental exercise, just like your Scrabble game, or that is for the spellings, or for the crossword puzzle, or the numbers. So this type of exercise, if you make, that would try to consolidate your memory stronger. The more thing important is you should not be stressed. If you a stressful mind cannot accept anything, I'm telling you. So that means the mind should be de-stressed without any stress. Okay, so now these are four components I mentioned for consolidation as well as storage. Now I have made one more thing here, the sleep. Normal sleep is important for memory consolidation and memory storage. The normal sleep, especially the REM, rapid eye movement sleep, is essential for consolidating the newly learned areas. So that means sleep is also important for normal uh, consolidation and storage of memory. So that means you try to, whenever what you do is during the time of examination, you don't sleep at all, and you just try to rot things, and you try to come here, and in front of the examiner, suddenly you lose everything. You don't get anything. No strings are coming up, and you say that, sir, I have mind block. That's a very interesting answer we receive from you or your seniors. So that means the proper sleep is also, because the sleep has a physiological role to consolidate the memory and store it properly, so that without any conflict, so the proper places, and without conflicting, without making compounding these things, they store well. So these are another interesting thing I did mention the exercise. I told about the mental exercises. The physical exercises also help the consolidation and the storage of the memory. Say for example, you take a break. So you just read a certain aspect. Take a break. Go for a small walk. That's even I'm not taking that going for a rigorous exercise. Just go for a walk or some run or something. So that exercise that tries to change of place, change of these things with inputs, the sensory inputs. You try to imagine about these things, and you will store and consolidate the memory very nicely. So this is about the storage and consolidation. Then about the retrieval. Retrieval depends upon, I have already mentioned, the cues you have stored the memory and the emotion associated with the memory. And this would be wonderful if you are calm, if you are not stressed, if you are not anxious. Okay, so this is where it becomes very essential when you have been facing the oral examination. In the oral examination, what happens? You become nervous, and you are anxious, and you are excited. So in that situation, it becomes difficult. Even whatever the subjects you may be knowing normally, under normal circumstances, you may not be able to bring. It is not your problem. The problem is here with the retrieval. So now the retrieval will not be proper. For that reason, you should have the calmness and distressing. These are the factors for the memory. Now, another thing, so this, when you retrieve a particular information, what happens? There are two benefits. It again comes back and try to consolidate. So that means it becomes a strong storage. A storage becomes a very much stronger. The more and more you do, the more and more it becomes fine, and you will be more comprehending about that particular topic. So now when you retrieve, it will improve the encoding, it will improve the consolidation, and it will improve the storage. So that is what, so this is the process of sequence of the memory formation. Okay, so now move on to this. The long-term memory, so just I want to mention here, the encoding process for short-term explicit memory involves the hippocampus. I have already mentioned about it. The memories are stored in various parts of the neocortex. I have already mentioned. Those are the parts: visual, auditory, olfactory, somatosensory areas. These information tied together for long-term changes in the strength of transmission, the transmission in the sense the synaptic transmission. So now for all these things you need to have the consciousness when the memory is recalled. So that means you should have to be attentive, and that attentiveness stores them as well as brings them back. Once long-term memories have been established, they can be recalled and accessed by many different associations. I mentioned each memory have 3 or 4 associations. That means somatosensory, visual, auditory, olfactory, or whatever. So for example, memory of a what I will say, vivid scene can be evoked not only by a similar scene but also sound or smell associated with the scene, and by the words such as scene, vivid, view. Thus each of the stored memory must be multiple roots keys. Just for example, if a cutlery sound is there in the kitchen, so that what does it, so I'm just trying to explain this phenomena. A cutlery sound in the kitchen that creates what you feel that food is ready, the plate is being served, and you try to imagine what type of food. Maybe your cook or a mother must have told that today I am preparing your favorite dish. So then that favorite dish comes to you. So like that. So the association of all these activities, and now also along with that you may have a smell. I've just said the cutlery sound. Now I'm talking about the smell coming, emanating from the kitchen. So that all adds to this thing. So that will be this thing. Now, the fourth component is an emotional component. So the smell of the food is good. So that is an emotion. Good is emotion. You will be seeing that, you will be excited that, oh, what a wonderful dish I have today, and I would enjoy. So that means it may be a pleasant one or unpleasant one, whatever. So that means these 4 components or 4 or 5 components put together, your memory makes memorable. On the other hand, your memory is stored. So that means any one of the incidents can bring in the memory out. Now, fifth one. Now, what is the neural basis of the memory? So now I will talk about the neural and molecular basis of the memory now. The key to memory is the alteration in the strength of synaptic contacts. Say, for example, what is this all about? It is only the neuron to neuron interaction. This neuron to neuron interaction is through the synapses. So now the memory is stored in the synapse. Synapse either in the hippocampus, amygdala, or somatosensory cortex, or prefrontal cortex, or wherever. So now the synaptic, it is the strength of the synaptic contact which tries to store the memory. Now these synaptic contacts have certain basic things. One, they are activated by ion channels. They are being activated. They involve the second messenger systems. So that means you have the second messenger systems that contributes for the strength of the memory. So that for the learning and memory, they become important. So then the ion channels, second messenger system leads to the working of the ion channels. The ion channels they become important for the release of the transmitters or for the action of the transmitters. So they are correlated with the learning and memory. The alteration then the memory and learning involves the synthesis of the new proteins, that is for the neurotransmission, for the ion channel expressions, or the receptor expression, for the neurogenesis, and so on. So that means for the synthesis of the new proteins, that genetic expression, and the activation of genes. This occurs during the change from short-term or working memory to long-term memory. So this whole thing happens when this is being consolidated from short-term memory to long-term memory. That is the crux of the, or the importance of the basis of the neural or molecular basis of the memory formation. Now we move on to this thing. So we say a word, this all things, whatever I mentioned in the previous slide, whatever I mentioned in the previous slide, they can be placed in one single or two words like synaptic plasticity. Synaptic plasticity means it is the adaptation of the synapses for the learning. So now what happens? There are short-term and long-term changes in the synaptic functions that happens associated with the memory, and especially in terms of the storage and consolidation. That means encoding, consolidation, and storage require these synaptic plasticity. What are these synaptic plasticity? So that means the synaptic transmission is strengthened, it becomes powerful, it becomes sensitized. So, or sometimes synaptic transmission is weakened or eliminated. So this plasticity is both strengthening and weakening or eliminating particular neurons, particular synapses, or adding or enhancing the synaptic activity there. So now this is depends upon the experience or the requirements. Now I've just put certain points here. Synaptic plasticity include the following: the formation of new synapses, then the efficiency of the transmission. That means whatever the synapse we have, the synapse, you have a greater that means the greater transmitter release. Just I mentioned the calcium accumulation and sequestration, then intracellular messenger amplifications, and second messenger signaling modulation. That means they are increased. Then gene expressions, so that signaling the receptor genesis or neurotransmitter genesis or a new protein synthesis, increasing the number of vesicles and in the presynaptic terminal, increasing the neurotransmitter content and release, the efficiency of the release, increasing the expression of the ion channels and neurotransmitter receptors. So that means we have in the synaptic plasticity, the synapse become more efficient. It has a more number of neurotransmitters stored in the vesicles, and it can release it faster. It has become active or it has become efficient. And finally, in the postsynaptic site, there are more receptors and more powerful the response. Now let us examine that. So okay, so just I have mentioned about synaptic plasticity. This may be one of the things our examiners are interested to know about. So now come back here, then go to this thing. So then in the synaptic plasticity, this becomes one of them, the long-term potentiation. Increased wave of depolarization after a stimulus in the synapse. There is a, after in the synaptic transmission, you have a post the excitatory post synaptic potential. Now the excitatory postsynaptic potential is a wave, it's a wave, a depolarization wave, and this excitatory postsynaptic potential has a time period. Now this excitatory postsynaptic potential may be prolonged. So this prolongation of the excitatory postsynaptic potential is known as a long-term potentiation. That means it becomes a long, usually it drops down within a few milliseconds to second. So the postsynaptic potentials are short-lived, whereas in case of a learning, there is a long-term potentiation of this depolarization. Or in case of inhibition, there is a long-term depression of the potentials. That indicates about the learning and memory. So that means if the depression of the inhibitory neurons may help to learn certain components, excitation or increase in the depolarization of the excitatory neurons may learn certain things. So like that, there is a balance of long-term potentiation and a long-term depression. So this long-term potentiation and long-term depression can happen presynaptically, that is the presynaptic terminal, or postsynaptically, or on the postsynaptic membrane. So these will result in increased activity after a tetanic stimulation. Tetanic stimulation means if we stimulate a particular thing with a greater frequency, that is a tetanus. So there will be normally the post in this event, there will be a greater response in terms of the LTP. That is there is a post-tetanic potentiation. Or in case of a LTD, there is a long-term depression, there is a suppression of the post-tetanic potentiation. These responses last for a minute to sometimes very long, even greater than a minute, 60 seconds or more. Now we move further. This is one of the experiment for the long-term potentiation. I've taken it from Kendall there, Kendall's textbook of neuroscience. So this is one of the classical experiments from the hippocampal slices. This is hippocampal slice. So now in this hippocampal slice, I will not go into the details of the hippocampal slice. So this is the recording electrode placed in the CA1 region. There are in the hippocampal slice, we have a CA1 region, CA2 region, and CA3 regions. So now the neurons, the pyramidal cell here in the CA1 region, it has been impaled, and the recording are surrounding that. We have placed a recording. And when we have placed the recording electrode here and it is connected to the recording device, maybe oscilloscope. Now in this area, we place the another electrode, so that would be stimulating electrode. When you stimulate these, this is a safer collateral. So this is a collateral coming from the CA3 pyramidal neuron there, safer collateral, which impinges upon the postsynaptic. This is the postsynaptic these things, and how are the presynaptic these things. This is the axon going out there. So now if when you stimulate this, so this would make a synaptic contact here, and how that will change. So that is what we see, and see this is what happens in the first instance. When you stimulated that with a particular train of pulses, you get the response something like that. This is the wave of depolarization. This is the wave of depolarization. This is EPSP slope, and this is going up and coming down, something like that. So that is the first time. And if you have a train of depolarization repeated or increased frequency, what happens? It would become longer and longer. See, at the fourth event and in the fourth attempt, it has become very long. So with the 4 trains, this is only one train. That means one frequency of one set of stimuli. And 2 trains is this much, and it's going on. It is not for purposes of clarity. It will go something like this. And in the last 4 trains, you get the depolarization there, increased depolarization, and this is sustained for several minutes. So see, look here, this is several minutes, 210 minutes. So that means nearly 4 hours. 64 to 40 minutes. So that means nearly 4 hours. It is this is a long-term depolarization. So now this is what the experiment, and I'm trying to make you to feel, to elicit early STP single train. This is early STP, and to elicit the late these things, the 4 trains are given separated by 10 minutes. 1, 2, 3, 10 minutes. 4 trains are given, 1, 2, 3, 4, and you get the after the 4th train you get a late LTP. So this is an early LTP and a late LTP. So this would last for 2 to 3 hours, whereas the first one lasts for 2 to 3 hours. This is the amount of depolarization, whereas the late LTP lasts for 24 hours or more. Now, what is that? That LTP. So now this is where I am coming back here. The possible mechanism of the synaptic plasticity, or LTP. So that LTP results in the changes in the neuron, the next neuron which its contact. So now I just read out these possible mechanisms of synaptic plasticity. The increased neurotransmitter quantum, increased receptive area, or both. Let us see what happens? This is the untrained or before training. This is the synaptic, this same experiment, what has been done for the hippocampal slice. So you get the transmitters, maybe 3 moles of transmitter released, and you this is the way of the postsynaptic potential. So this is the way of depolarization. Okay, this is where it is recorded. So now what happened here after training, that is the memory. So now the training is the memory in which you see the quantity of transmitter is increased. So you have 5 moles. This is 3 moles here. The number of neurotransmitter molecules are increased. That is one. That is what I mentioned here. Neurotransmitter quantum is increased. So now what happens? Even if it is 3, or even if it is only 3 moles of these things, you just see this postsynaptic site. This postsynaptic site has the receptors. Now the receptor area is increased. The number of receptors are increased. So that means this is effectively, these 3 moles effectively interact with the receptor area. So that means that would produce a greater response. In this situation, maybe 1 or 2 moles may be activating. All the 3 would be able to activate the receptors. And the third thing is, so it is both. So there is an increased the quantum of neurotransmitter release, increased the area of a receptor or postsynaptic site. So what at the end you get the increased postsynaptic potential and increased long-term potential. So this is what happens in case of long-term potentiation. Now then we will go with the next these things. This again the mechanisms for the synaptic plasticity. This already we are knowing about this. So this is a postsynaptic potential. This has increased the area as well as the transmitter released. So that's one component. The second component, what's happening? It will try to make the new synapses. Say for example, another dendrites is coming up there. So now after some time, after repetition, after rehearsing, after these things, you have another synapse formed. You have two synapse formed. So now these 2 synapses are activated. So that means a new synapse formation takes place. Then third component, rearrangement of the, say for example, we have the synaptic contacts here in the beginning, synaptic contacts here when it is stimulated. Now this would this stimulation will this would see, for example, this is unused. This is unused. So now this is snipped off, and this will take this synapse, and this is the rearrangement of the synaptic input. So this is the neuronal plasticity. I'm trying to explain all those things. Long-term potentiation initiates all these changes cellularly, involving the genetic expression of a new protein, because this is you require a new protein there. That means you have to eliminate this connection. You have to increase the vesicle synthesis. You have to increase the receptors. So all these things require a protein synthesis. That is what the synaptic plasticity is about. Then one more interesting thing you see. So now we have seen the greater vesicle release for neurotransmitter secretion. The greater number of transmitter vesicles are released. Then greater number of presynaptic terminals I have mentioned. So see, for example, this is a untrained. So this is learning. Learning what happens? The new dendrite. This is a dendrite. This is a dendrite. New dendrites are being formed. New dendrites are being formed. So if you are looking at this, there are only 3 here. There are 4 coming up. There are 5 here. There are 8 coming up. So that means new dendrites are forming up. When this particular neuron gets stabilized, you see that there are at the end 5. So the 5 number, they will be stable, and then you have the memory stored in this area. So that means the stabilization of the activated synapses that happens. That means it brings the structural changes in the neuron, especially in the dendrites. They increase the dendritic arborizations, and these are called dendritic spines. The dendritic spines, where these spines make a contact with the another synaptic button there. So more synapses are formed. So long-term potentiation tries to bring in the changes in the neuron. Now we move on. The memory and the neurogenesis. The traditional view that brain cells are not added after birth. That is what we believe. Now it is not true, because certain areas in the brain, there are neurons which are still able to divide. Especially new neurons could be formed from the olfactory bulb and some parts of the hippocampus. So these are the storehouse wherein they will produce a new set of synapses, or new neurons are developed. Now, in this context, if I try to bring in you a person who is there, and with an accident he loses his eye, and when he becomes a blind, so the entire visual cortex is not able to receive the visual information. In that way, he develops other skills such as tactile skills. He tries to understand the entire world either to the touch or from the sound. So the tactile or the auditory signals, they occupy that space, because the visual area, the occipital area now is not used, or not used because there are no signals coming up. So that is a neuronal plasticity. Neuronal plasticity, the tactile processing of information in a person with a blind, or a person with a blind is, you must have seen many of the musicians are blind. They are wonderful musicians. So it's in, that means there is a area of the that area of the cortex is being used by the other sensory modality, which is important for the survival. So that means another important aspect. So if you see an area, say for example the Hessel's gyrus or auditory cortex, it will be very large in case of a musicians of established level, of high level, or the persons, the great painters, they have a somatosensory, the hand skill movements, they have a large area, the greater area they take up. Or if you tomorrow, if you try to become a great surgeon, and that is again a skill, though your area will expand. Or as a teacher, maybe if I have recorded my teaching abilities when I started 40 years back, and now maybe I could have expanded, or in my brain they expanded the area of representation. So this is called a neuronal plasticity. So new neurons form from the stem cell throughout life. At least they are originating from olfactory area and hippocampus. This process is called a neurogenesis. Experience dependent growth of the new granule cells in the dentate gyrus. Dentate gyrus is connected with hippocampus, and that will be contributing to the learning and memory. A reduction in number of new neurons formed reduces at least one form of hippocampal memory production. Okay, we will move on. The neuronal plasticity, I have talked about the synaptic plasticity. Now I am talking about the neuronal plasticity. It is the increased number of neurons or glia, as I mentioned, for a blind or a losing particular area. So increased number of dendrites and spines of the dendritic spines. The new synapse formation, then synaptic plasticity, the efficient synaptic plasticity. Already I mentioned all these components, the formation, increased accumulation, efficiency, neurotransmitter content, the ion channels, the vesicles, the postsynaptic receptors and ion channels, intracellular messenger amplifications. So all these things happen in neuronal plasticity. Now, the disorders, let us consider the disorders of memory. The memory disorders include the amnesia and dementia. The amnesia is a loss of memory. So I have already mentioned about the anterograde amnesia when I was talking about the amygdala and the hippocampus or the limbic system. So in this situation, they are unable to retrieve they are unable to form any new memories. No new memories are formed, but they have the intact the previous memories. Retrograde amnesia, on the other hand, I mentioned about. So there is a loss of previously stored memory. So that means amnesia, loss of memory. There are two types of amnesia: anterograde amnesia and retrograde amnesia. Now, Korsakoff syndrome, I have talked. It is a loss or the abnormality or lesions in the mammillothalamic tract, and this may have both. This may have both anterograde and retrograde, and this is seen with the alcoholism and the thiamine deficiency in case of the malnutrition. So now the third part is the dementia. Dementia is a broad category. We have a neurodegenerative diseases. One of the greatest neurodegenerative diseases is the Alzheimer's disease. Now, one more thing you may want to ask about, which I have not typed, the forgetfulness. Forgetfulness is it happens with everybody at any age. So it depends upon the context in which we are making. So that means the forgetfulness is a part of the memory development, and sometimes we tend to forget certain issues, we tend to remember certain issues, and finally it is a formation and the consolidation and the storage component. Now, coming back with Alzheimer's disease, because it's one of the question, I just quickly read through these things. It's a most common age-related neurodegenerative disorders, happens in the elderly people. The memory decline usually manifests loss of a short-term memory, especially the episodic memory. The person goes for a walk and he does not find his house back. So that means he will not be able to come back to his house, and somebody has to help. Loss of short-term memory followed by general loss of cognitive and other brain. That means the intellectual component is also lost and other functions are also lost. Manifest with the agitation, depression, and a constant care and need constant care, and eventually death. Because they will not be able to eat. They don't know whether they have eaten or not. They will say that I have eaten, but they don't eat. Or they may keep eating sometimes because they don't remember anything. So this is because of the loss of cholinergic neurons that connects the nucleus basalis of the main heart of the basal forebrain. The diffuse cholinergic projection goes to entire neocortex and amygdala, and thus there is a grave disease. So this is one of the things. Maybe with all your elders there, your great grandmother or something like that, they may be trying to recall you, and you may say that I am a daughter or a son of somebody. So then she will not be able to recall, and suddenly she may recall, and again she may ask, keep on doing like that. Now, what is the problem there in Alzheimer's disease? There are what are called neurofibrillary triangles. These are neurofibrillary triangles. So it's a neuron I have drawn there. This is the neuron. You have the neurofibrillary triangles, which are made up of part of the hyperphosphorylated tau protein, which is synthesized there, and that normally binds to microtubules. That is, the microtubule is the one which transmits the neurotransmitter synthesized in the soma. The tau protein, so that is hyperphosphorylated, and it becomes condensed, and it becomes precipitated there, and they make the tangles. The extracellular, in addition, there is a formation of a protein in the extracellular component. So this extracellular amyloid plaques, these are amyloid plaques, these beta amyloid peptide, so they are there, and they are synthesized by the glial cells. They produced are surrounded by altered nerve fibers and glial cells, and they produce a inflammatory reaction. And especially these peptides, whatever the beta amyloid peptides, they are really toxic. Whatever they have produced, they produce a toxic events to the cell, and they kill the neurons. Maybe they kill the neurons, and that is what is the important thing that beta amyloid peptide produces amyloid precursor protein, and this will produce the toxic chemical, or these polypeptides are toxic to the cell, and many of the neurons will die. And once they are at the same time, the neurofibrillary plaques, the plaques, they are produced, the amyloid beta plaques. So these produce they aggregate, and they will activate the AMPA receptors, that's a glutamate receptor, and that glutamate receptors, and also the calcium ion channels, so the calcium influx. The calcium influx results in excitotoxicity and again cellular death. So that means by two counts, the cells are, or neuronal cells are killed or dying. So that results in the loss of the neurons, and that is where, and in addition, there is inflammatory response, and this is what happens in case of Alzheimer's disease. This is a healthy brain. You have so many gyri and sulci. You can just see that it's so nice, and you see that it is all smoothened brain, and it has become smaller because there is a loss of the neuronal cells. This is an advanced Alzheimer's brain on this side, and you have a healthy brain on this side. You can just compare the number of neurons. So that is what happens in case of Alzheimer's disease. Now, just to give you an idea how Alzheimer's disease could be treated, the only treatment is see that they don't advance it fast. There is a slow progression, and help them with whatever the symptoms they are having. So that means you require a good nursing care. You have to take care of this patient because they are forgetful. They don't remember anything. So you have to care them. And so you try to help their cognitive activities by asking them to solve certain puzzles, or playing board games or whatever. So that means keep them mentally active. That's the second component, because these require nothing. But it requires attention. So 100% time, it's very difficult to have these attention kept with these Alzheimer's patients. So now the drugs are, because there is a cholinergic neurons are lost, you can use the anticholinesterase inhibitors, may help to relieve, but it may happen in case of in the beginning, but when all the neurons are lost, so this one anticholinesterase may not help, because there is no acetylcholine coming up. And then the memantine is a NMDA receptor, because there is a balance between the cholinergic and the glutamatergic transmission. So now here you can use the glutamatergic because increased activation of the glutamatergic transmission, that is leading to the excitotoxicity or any other damage it is producing. So that means you try to decrease the glutamate activity through the NMDA receptor antagonist memantine. So now still we have the drugs which can block the beta amyloid protein. They are under development, and the vaccines are under progress. That is what the Alzheimer's disease. Now, seventh is the memory enhancement strategies. I have listed here, maybe I've taken it from all the studies. There are three components. Maybe it will interest you for yourself. One, lifestyle strategies. Two, the beverages and herbs, what we call it as decoctions or some herbal preparations, and the memory enhancement drugs. Memory enhancement drugs are called nootropic drugs. Now, the lifestyle strategies. So that means you increase your attention level, alertness or attention level. That is one point. So when you want to have a good memory, you be attentive. The second is you fix. It is self-driven. The memory is to be achieved by self-driven. It cannot be forced upon by the teachers or the parents or somebody else. You have to be self-driven to learn and try to remember. So that means you have a goal, you fix a goal for yourself and create an interest in that area and motivate yourself strongly, and that you will achieve you can increase your memory. So then third thing, maintain proper sleep duration. because the sleep I mentioned, besides many other functions, it is necessary for consolidation uh, and storage of the memory. So for that reason, you should have a good REM sleep for memory consolidation. The fourth thing is you rehearse and repeat at frequent intervals so that you stabilize your memory, you consolidate your memory, and it's stored well. And you always associate with these things with the cues, with some emotional cues. Maybe that would be stronger to keep the memory intact. So then have a proper physical and mental exercises, trying to develop a quiz for a particular pattern, or if there are any quiz for a particular level of learning, you use those quizzes because that would enhance your memory, or that would keep you storing, consolidating that memory better. Then the meditation, bringing up the concentration, that is focusing, and introversion and prayer. So these are all one. It is a sort of a spiritual level. So, but they try to calm you down. For while I mentioned in the retrieval, there is a calming. Calming is necessary for to retrieve the stored information. So that means these also help to retrieve or even to consolidate and store the memory. And while learning, try to have a space between the learning schedule. So don't cram it or don't push it too much. The brain cannot take anything. There is what is called an auto shut mechanism. The brain shuts down. That's all. You don't. You want to read. You are reading. Your eyes are seeing the words. The maybe that's all. It may not be storing. So that means you have a space or a time period between the learning schedule. That is one. Then herbs and beverages. We have caffeine. Caffeine increases the alertness and concentration. The caffeine is there in coffee and teas. So that would be better, so that you want to have the learning schedules. Maybe caffeine would help to keep yourself alert, and thus it helps to store the memory or consolidate the memory. L-theanine is present in tea, and this has a calming effect. The calming effect is without drowsiness. So that means the caffeine is also present in tea, theanine is also present in tea. So that means tea has a double effect in terms of the working for the memory. So that means it calms you down, it cools you down, and so that your consolidation, retrieval, rehearsal may be easier. I would not advise for the nicotine. It's an addict forming drug, but it would stimulate and it will keep you alert, but I would not say that. So now creatine is a thing, a dietary or a supplement which will increase the formation of the ATP, like a phosphocreatine, which will increase the energy levels. So that may help to increase the memory, because the energy levels are required for the brain. So then we have a Brahmi. So Brahmi preparations, so we have a Bekopa Muniri. So these are Ayurvedic preparations. So we have Ashwagandha, and we have other Arjun, and many other Ayurvedic preparations are coming up. The Brahmi is one of them, is Ashwagandha preparation. So that has an alkaloid that would increase the memory. So that is how they claim, but the experimentally we don't know, but it may help. So then we have a Rhodelia rosea, that is another preparation, the Ayurvedic preparation. This is adaptogenic. Adaptogenic means it diminishes the stress levels. Diminishes the stress levels. By diminishing the stress level, your learning is enhanced. Then we have a ginseng. Ginseng is a sort of a root, and again it's a medicinal plant that is mentioned to be the the brain function or especially the memory. So now you have a ginkgo biloba alkaloids. It's a Chinese herb that will improve the memory and the mental processing and may be beneficial in stressful situations. These are some things I am just giving you broadly. So details, one is each one of them are the areas by themselves. Then what are the nootropic drugs are which increase these things? These are some of the drugs which have been approved by the FDA. So I would not go into the detail. I have already mentioned about the fast acting synthetic nootropic that may improve memory by increasing BDNF levels in your brain. That is donazepam, rivastigmine tartrate, galantamine, memantine, NMDA antagonist. So we have a psychostimulant, modafinil, and amphetamines. Once a time, amphetamines were prescribed. The problem with amphetamines is they will keep you active. At the same time, the memory regained cannot be recalled or retrieved. So that is the problem. So that will become sequestrated. So now we have a methylphenidate. So these are some of the nootropic drugs. I'm not going into the detail. I have just listed them for your interest. Okay, with these things, I have covered the entire topic of the memory and the learning, and these are some of the keywords: the memory and learning, the types of memory, long-term potentiation, the classical conditioning versus operant conditioning, stages of memory formation, sensitization and habituation, Alzheimer's disease, short-term memory, working memory, episodic memory versus semantic memory, and explicit memory versus implicit memory, processing of memory, synaptic plasticity, other lifetime. This is a lifetime changes. The changes in the how to improve the memory enhancement procedures. You try to recall all that. So these are some of the reference books, and in addition, I have referred many more books which I cannot list all of them here. Okay, thank you.